traditional custodians of the lands that you are all joining me from today, and I pay my respect to their elders past, present, and emerging, and to the Aboriginal elders of other communities. So welcome everyone, my name is Anya and I will be chairing the seminar today. I'm a PhD student of the CCMP Center at the Monash Node, and before we start and introduce our speaker, I would like to quickly go over some housekeeping rules. So first and foremost, I'd like to let you know that today's seminar will be recorded and you will be able to find this as well as all the previous recordings on the CCMP website. And the presentation by our speaker will be 45 minutes, followed by 10 to 15-minute Q&A session. So please leave your questions till the end of the seminar, either by raising your hand or typing them in the chat box, so we can all, of course, address them later. Please also make sure that your microphone is muted throughout the presentation. So our speaker today is assistant professor from the University of California, Kliment Verba. He obtained his PhD in Dr. David Agard's laboratory, working on Cryo-EM studies of kinase chaperone protein complexes. After his PhD, he received an independent research fellowship at QBI UCSF, starting his own lab with the focus on structural studies of near-membrane kinase signaling. His recent work helped uncover principles of asymmetric regulation in aGFR family receptors, and also how such signaling propagates downstream through cytosolic protein complexes. So during the pandemic, Dr. Verba helped organize a large volunteer effort focused on structural studies of SARS-CoV-2 host viral protein complexes, providing structural snapshots of a number of these potentially druggable complexes. Lastly, he has contributed to novel methods in developments in cryo-EM data collection and processing. So his talk today will be about using the power of cryo-EM to uncover principles of kinase signaling and the membrane. So over to you, Professor. We are very lucky to have you here today, and the screen is all yours.
Great. Thank you so much for the introduction. Let me share my screen. Let's see. Well, thank you so much for that introduction. And thanks everyone for tuning in. And yeah, so today I'll be telling you about using cryo-EM to uncover principles of kinase signaling at the membrane. And so fundamentally, my lab is really interested in how cells make decisions. And so as we all know, cells have to read in inputs from the outside of cells, be it from growth factors from other cells or hormones. Especially this is incredibly important once cells decide to form mini societies and multicellular organisms. They need to incorporate and integrate those inputs from the outside of cells with their internal states. So they need to figure out, where they are in their own life, how damaged their DNA is, how many nutrients they have, and then ultimately they make a decision. Do they divide? Do they differentiate? Does it come into apoptosis, or do they move around? And there has been this has been studied for a very long time. And a really interesting example, well, a classical example system of this is PC12 rat cells, where the same cells, if they're treated with EGF, they just divide. And if you play this movie for longer, you'll see them just dividing. But then if you take the same cells and treat them with NGF, you see that they very rapidly differentiate rather than divide. And so as a structural biologist, for my lab, the goal is to understand the molecular mechanisms of this decision making. So what do we do? What do we know about this decision making? So what we know is that a lot of the decisions are made by protein kinases. And so here we see sort of from a review, we see a diagram focusing on the eGFR family of kinases, shown here in green, which are receptor tyrosine kinases, which are in the membrane. And so there is a primary unit which will take the extracellular signals, like growth factors, and then that'll convert them into intracellular signals. But then right below this growth factors in green, not growth factors, but receptor tyrosine kinases, which are responsible for binding to the growth factors, we have the signal processing layer, which also has a lot of kinases in blue over here, together with other enzymes and scaffolding proteins, which are responsible for integrating that signaling from the receptor tyrosine kinase, and ultimately converging on the transcription factors to change the cellular programs of the cells. And so in the lab, we've been really picking apart at this. We take a holistic view, sort of a structural biology meets system biology view of the signaling, where Allison and Brendan, for example, have been really working hard on this HER3, HER2 containing protein complexes. Komal Pawar, postdoc in the lab, has been really working on this, trying to structurally understand how this module over here works, the RAS, RAF, MEK, ERK module. And I'll talk briefly about some of the progress she has. And then Hayarpi Terasyan, a grad student in the lab, she has been working on, amongst other things, has got a really cool recent story on a really cool pseudokinase, PEAK3, which is downstream from EGFR signaling, EGFR receptor tyrosine kinase, and how that integrates with 14-3 proteins which recognize and change the complexes based on the phosphorylation state of the pseudokinase. And Lisa, a grad student in the lab, she actually does not work on she's the only one who doesn't work on protein kinases, but works on MYC, a transcription factor which is intrinsically disordered. Actually, she works on NMYC, an isoform of MYC, well, homolog of MYC, which is a downstream receiving unit of all of the signaling from the kinases. But what I'll really talk in quite more depth today is a work which was done by Devon and Raf on structural studies of HER2, HER3 receptor tyrosine kinases in their near full-length context, which is depicted over here. And I would like to point out that pretty much all of the kinase work, which I'll be telling you about, is been done in a close collaboration with Natalia Ewer, and particularly this pertains to this HER2, HER3 story I'll be telling you about. It's something that Natalia has been working on quite a long time alone before I joined UCSF. So if you zoom in a little bit on this HER2, HER3 signaling in the EGFR family, we need to first understand a little bit better what's the canonical model of EGFR signaling is. And so EGFR family receptors, they're a single pass transmembrane receptors, and they have this ECD, extracellular domain, which is composed of 4 domains, and a kinase domain on the intracellular side. And then upon growth factor binding, these extracellular domains, as you can see here, upon binding to the growth factors, undergo large conformational changes to dimerize. And this dimerization extracellularly leads to further downstream dimerization, and there's plenty of evidence to also further oligomerization, which leads to at least well, one way of downstream signaling is through this kinase dimer, where you get this one activator, this one kinase playing an activator role, and one kinase domain playing the actual active kinase, which then leads to phosphorylation of this disordered tails. And these tails can be quite long. They can be 200, 250 amino acids. And one of the key elements of this activation upon growth factor binding is there is this depicted here, this little triangles, as this dimerization arms, which really sort of swap around between the 2 receptors and then really stabilize this complex. And so this is a canonical model which have been arrived at through a myriad of structural and biochemical studies in the field. Really prototypical family member is EGFR. But the EGFR family contains 4 receptors, receptor tyrosine kinases. And so we talked briefly about the EGFR. There is HER4, which is thought to really signal and behave very similar to EGFR, where it goes from this is called a tethered conformation, to the extended conformation upon growth factor binding. But then now these 2 other members here, HER2 and HER3, are very curious. And so HER2, depicted here in this sort of orange color, it's been the extracellular domain has been captured structurally through crystallography and actually Cryo-EM. And it's always been seen in this extended conformation. And so there's no known ligands for HER2, and it's thought to it does not require ligand binding for it to be in this sort of on conformation, seemingly always ready to dimerize. And then HER3, the extracellular domain, is thought to behave very similar to the aGFR, but then the kinase domain is actually a pseudokinase, so it lacks a key catalytic residues and is thought to not possess any catalytic activity of its own. But furthermore, what's been well established is that there is some of the worst breast cancer cases with worst prognosis are driven by HER2 amplifications or mutations, and often this HER2 signals through this obligatory heterodimerization with HER3. And so this particular pair between HER2 and HER3 is a very strongly oncogenic signaling pair across the family. Of course, the aGFR itself is already very well established as an oncodriver, but then HER2, HER3, because HER3 has multiple phosphorylation sites which can directly recruit PI3K downstream signaling molecule. And then therefore, this is a very potent oncogenic signaling union. However, although we seem to know quite a bit about this, as I've been telling you, all our structural mechanistic understanding has really been based on domains. And so, for example, we have aGFR and HER4 crystal structures of their tethered extracellular domains. We have crystal structures of this symmetric and asymmetric extracellular in extended conformations bound to the growth factors, as you can see over here. We have NMR structures of just the transmembrane helix dimers. And then, of course, we have this kinase domain dimer, which Natalia actually solved, where you can see one kinase activating one kinase domain, activating the other kinase domain. And so to date, there's still no full-length structure of either aGFR family receptor, or actually there is any other receptor tyrosine kinase. So although there have been many efforts, especially recently, we've as of yet to visualize a full-length receptor tyrosine kinase, both extracellular, intracellular, and transmembrane in high resolution. And so this leaves many questions open. How, for example, as you can see here, HER2 does not need to bind to extracellular to any growth factors. What prevents it from being constitutively active? Again, HER3 has been crystallographically captured the extracellular domain in this tethered conformation. It's thought to behave the same way as, say, HER4, aGFR in the extended conformation, but that has as of yet to be shown. And then, so how does HER3 in the on state look like? And of course, again, all the structural work has really been either HER4 homodimers or aGFR homodimers. And so how this potential, very strong oncogenic unit would look like, the asymmetric complex has been unknown. And probably the biggest question really is, is the total greater than the sum of its parts? As you can see here, through decades of very intense work, we have very detailed studies of all kinds of different domains and how they interact with each other. But is there some things that we're missing from by not seeing the whole picture? And so this sort of set up as they laid down the groundwork for the overall project, with the goal of really trying to understand near full length context, the structure for HER3. And there have been a number of enabling technologies which really allowed us to do this work. And so one of them is really using this EXPI 293 HEK cells, and so which are suspension cells. They're commercially available, so hopefully all of you guys are using them. But the coolest thing about them is the fact that they can grow to incredibly high cell counts. I don't think we really get in our, with the samples that quite that high, but we get into the tens of millions per mil. And so 20 million per mil of cells is about one 15-centimeter dish at confluency of adherent cell culture. And so realistically, by going into this 30 mil cell culture, or even we mostly grow, 100 to 200 mils, allows us to really get quite a bit of biological material to start our work with. Another really cool benefit of working in the system as compared to, say, even insect cells, even though insect cells are pretty good also, is the fact that if you're missing a protein, if that protein is in high abundance, you should be able to actually pull it down too, and then you can follow down mass spectrometry to identify the protein, and then you can overexpress it. And so realistically, if your protein complex requires a third member, which you are unaware of, using this physiologically relevant system allows you to more often than not capture it. And so we see it routinely with 14-3 proteins, for example. And so using the cell system, Devon and Ralph, Devon started out, I remember, some of our first meetings, scraping 20 to 30 15 centimeter dishes to get like a very, very faint band, which, you really need to squint. But then going to the cells and using this particular construct, so HER2 and HER3. So these are not full lengths, they're truncated at the tail. So we only have short extension of the tails post the kinase domain. And we incorporate a couple of mutations. So there's this mutations which are known, previously been shown to stabilize the kinase dimers. And also there is another mutation in HER2, which makes it HSP19 dependent. But the purification is actually, I think, pretty cool. And so basically what we use neuregulin, which is a growth factor, which binds to HER3, conjugate to flag. And then we can use this neuregulin flag to conjugate to flag beads. And so as if we basically have a bunch of cells that are expressing HER3, bunch of cells that are expressing HER2, and then we co-lyse them and then pull on this conjugate neuregulin flag to the flag beads, and then pull on the complex as a first step affinity purification. And then we pull on MVP to pull on HER2 to get the heterodimer. And in the end, even from this fairly large cell count, we get very measly looking size exclusion peaks, but, it's good enough to see on the Kumasi, as a very clean protein. However, as you can see here, the absorbance is very low. And so one thing we really need to, we had to leverage quite a bit is using graphene oxide grids. And so this sample and the structure I'll be telling you about has been prepped in DDM mice cells. We actually tried DDM mice cells, GGN, digitonin. We tried going into LMNG over basically and doing a size exclusion over no detergent. But the problem, as probably most of you know, once you have DDM mice cells, you lose this sort of concentration ability, this effect that where your protein gets concentrated on the air-water interface, and it's because it's coated by the detergent, and therefore you need to go to quite high concentrations. And so when we started out, we would get like one particle in the whole, and we really, which is totally not workable for structural biology. And so using this homemade graphene oxide grids over here, which are really developed in the Ager lab, and Feng specialist in his lab really helped us a lot with this, allowed us to get really quite good sample coverage, even with such low yields. And so one cool thing about preparing these geo grids in-house rather than commercially purchased geo flakes from the Sigma, for example, is that the way Feng prepares them, it's a modified Hummer method, where you get this geo flakes as larger. And so when you code the grids over as in shown here, you actually don't have to, whenever you code the grids with graphene oxide, you always have this balance of how much of your grid is going to be undercoated and how much of your grid is going to be overcoated, because you get a distribution, because there's a multiple flake rather than a continuous single crystal of graphene, for example, as done in, say, Passmore, Chris Russer's lab. And so having bigger flakes allows you to sort of minimize the variability, where you get on average, 2 to 3 flakes on the grid, and you still get good coverage. If you have very small flakes, then you need to go up in your concentration of flakes, and you can get spaces which are very high, which will have very many flakes on top. And so being able to do that, and of course we collected large data sets on the Titan Krios here we have at UCSF, and processed it through sort of standard classifications and refinements, which I'm skipping. But at low resolution, we're able to get the structure depicted here, as you can see, which really sort of seems to encompass at least a very low resolution model of the full X receptor. And so you can see here the ECGs, here sort of the micelle with some transmembrane domains hopefully in there, and the kinase domains would be somewhere in this blob. But but we only been able, this is a very low resolution and low threshold values. However, at high resolution, we're able from the same reconstruction to capture the extracellular domains, as you can see here, at about 3 angstroms. And so what was really cool overall, they kind of looked similar to previously captured aGFR extracellular domains. But then when we zoom in at this key dimerization motifs, the dimerization arm, on HER2, you can clearly see dimerization arm depicted here in blue, very well resolved. But then on HER3, to our big surprise, we saw no dimerization arm density, as you can see here, where we had to cut it out, and there are absolutely no densities there. And so today, and both HER4 and all the aGFR structures, of which there are many, always both of the dimerization arms between the two receptors have been resolved. And so this was very, very unexpected from our work. And so we wanted to make sure that this was not some sort of processing artifact. And so, of course, as some of you know, in CryoSpark, there's automatic masking. And we want to make sure that it's not just being masked out because maybe it's a little bit weaker, which could be interesting and so on, right? And so we went ahead, and if you look at just the CryoSpark half maps, you can see that even in the half maps, which are unfiltered, they're unfiltered and unmasked, most importantly, you see that you still have this asymmetric dimerization arm. When HER2, you clearly see dimerization arm very well resolved, but on HER3, you really see no dimerization arm. Furthermore, we actually took this particle stack from CryoSpark and we imported into Reliant. And then we actually started out with a reconstruction which had symmetric dimerization arm density. So the initial model had 2 dimerization arms. And then we did a refinement on the particle stack using here a spherical mask. So therefore, you have no chances of masking out specifically dimerization arm. And you still have a dimerization arm missing. And then if you would do post-processing to get a little bit sharpening to get a little bit higher resolution map, but the mask still included both dimerization arms, you still saw that there was a symmetry in dimerization arms. So therefore, we're fairly confident that this is not an artifact of our processing. And of course, the only other thing is, a concern could be, was there processing in terms of, say, classification, where somehow we focused on the dimerization arm during 3D classification? And it's also not the case, because realistically, the only particles which we threw out were really obviously junk. We very closely examined every other class, and there are really no other classes which really looked very similar to the sort of heart-shaped dimer. And therefore, and also, the dimerization arm is about 1.5 kilodaltons. And so realistically, as whoever tried it will be able to tell you that being able to classify based on such a small feature would be very difficult. And so we really don't think that, we think this is physiologically relevant finding and not an artifact of our processing. But then to show it more unequivocally, we're actually took basically this would argue that HER3 dimerization arm does not matter for signaling through this heterodimer of HER2, HER3. And so what we went ahead and we replaced the dimerization arm with this just glycine serine linker. And then as you can see here, this mutant was able to activate and lead to phosphor HER3 levels, the same as a wild type, showing that indeed the dimerization arm of HER3 for binding and signaling through this heterodimer does not really matter. So how can, so now that we're reasonably confident that this dimerization arm is actually curious and not an artifact, how can we explain it? Well, of course, this is an N of 1, so we have to be careful because there could be just idiosyncrasies which are specific to this particular heterodimer system. But we think we can get, we hypothesize that this is actually a feature which is relevant to the rest of the aGFR family. And so if you look over here at our structures, this is just looking at this as the extracellular domains. And starting over here, this is a HER3, as you can see in gold with neuregulins, the ligand bound. And so now this movie will go forward and will actually morph from HER3 to a HER2 state. And so what you can see is that this subdomain 1 and subdomain 3 actually rotate between each other as you go from this HER3 state to the HER2 state, which is maybe a little bit seen a little bit better here. And so what you see structurally when you look at this is that actually what this interface over here, this region over here, is actually the region which receives the dimerization arm of the other receptor. And so what we think happens is that depending on your ligand and the state of your ligand being bound or not bound, you basically sets up that extracellular receptor to receive the dimerization arm of the other. And in the field, so this is called sort of wedging between the subdomain 1 and subdomain 3. And so what we see is that in HER3 state, which is neuregulin bound, which is very similar to sort of the canonical EGF bound aGFR, we see what's called this fully wedged conformation. And so basically neuregulin is wedging between the two as a subdomain 1, subdomain 3, and then it sets up the binding pocket, and all the contact residues are shown here in red, so that the dimerization arm can bind quite well. Now, on HER2, because HER2, remember, does not have a ligand, there's none of this wedging that happens. And so you end up with this very more open pocket. Now, if you look at the eGFR structures, and again, we think this potentially is, this is relevant beyond just a specific HER2, HER3 case, which we see here, is that the eGFR has this fully wedged, has been observed crystallographically in this fully wedged conformation with EGF as a ligand. And again, there in red, you see all the contact residues which interact with the dimerization arm. You can see a nice, well-ordered pocket, binding pocket for the dimerization arm. And then it's been observed in this subtly asymmetric state bound to ERAG, which is a weaker ligand. And you can see here that this is termed as a partially closed pocket and a partially wedged conformation. And you can see that this pocket now has less contact residues than the fully wedged. And so what we think that potentially is going on is that there is a spectrum of basically wedging depending on how engaged with the ligand you are. And depending on that sets up how well is your binding pocket set up for the dimerization arm of the other receptor to come in. And what was really cool to our analysis as we were going through this is when we looked at this, compared the existing crystal structures, this is not our structures, of EGFR EGF and EGFR ERAG, even though both of them resolve the dimerization arms and build them into the structures, the fully wedged conformation with EGF, the B factors were symmetric. But then EGFR ERAG structure, although both of the arms were built, there was asymmetry in the B factors, which sort of correlates with the asymmetry, extreme asymmetry we see with HER2 and HER3 now structure. And so we think that the potentially is this sort of, this lack of setting up of the binding pocket leading to destabilized dimerization arm interface could be actually, is an extreme case of a fundamental mechanism in the EGFR family. And so this has been thought about and observed for, again, this ERAG EGF in terms of thinking of it as being partial agonists, biased agonists, sorry, where you see that ERAG can actually lead to different signalings through the same molecule as EGF. And that has been thought to go through this different asymmetric state on ERAG. And so what we think is going on here is that HER2, HER3 basically show up the two extreme data points on that, where HER3 is fully wedged and then HER2 is completely unwedged. And this ERAG structure, as shown here, is partially wedged. And so if you compare this, again, the orientation between subdomain 1 and subdomain 3, we can see with ERAG, the rotation between the EGFR and HER2 would be 20 degrees, and between EGFR, EGF and HER2, we see the rotation would be 30 degrees. And so depending on what ligand comes in, so again, we have this asymmetric, very asymmetric structure, but we think that there are some fundamental ideas here which transfer to the rest of the EGFR family, where depending on which ligand comes in can change the degree of wedging, and that changes how good this binding pocket is set up for the other dimerization arm. Changing the strength of the interface. Okay, so we saw this dimerization arm, we did this analysis, which we thought were cool, but we can often see things in Cryo-EM which are disordered, which are not there. So, is this actually relevant? Is this good for anyone or not? And so Devin and Raf went into the cosmic database for cancer patient-associated cancer mutations. And HER2 usually is overexpressed. That's sort of the most, that's the most likely oncogenic event. But in terms of point mutation, there was this one which really jumped out at them, which was S310F. And what was really cool is that this S310F was actually in the region of HER2, which would be right where HER3 dimerization arm would be interacting. And so they got quite excited about it. They went ahead, put it in their mammalian cells, prepped it, purified it, And then we got the Cryo-EM structure of that. And so, and now if you look at that, if you compare the structures I got from this to the structure which I've been telling you about so far, we now actually were able to really resolve very well the dimerization arm of HER3 in this mutant, cancer mutant structure, which was really cool to us. And that was because of this phenylalanine was making pi stacking interactions with residues on the dimerization arm of the HER3. And so I think this is really actually quite cool, because basically this indicates that there's this finely tuned dynamics at the interface of this very potent oncogenically signaling heterodimer, which is finely tuned evolutionary to be not super stable. But then this cancer mutation, a single point mutation, can basically staple this dimerization arm, stabilizing this interface and driving disease. So that was very satisfactory to see. And lastly, it's been, Herceptin, because again, HER2 has been such a disease-relevant molecule. HER2, there's a number of inhibitors against it. And some of those inhibitors are monoclonal antibodies, FABs, like Herceptin, and trastuzumab and pertuzumab, as I also know. And so Devin and Raf wanted to see if we could capture a structure of our, now Herceptin has been captured bound to HER2 alone, but it never been visualized. There been actually some sort of discussions where it was unclear if it would actually bind to the complex of HER2, HER3, or if it would disrupt it, etc. And so when they added the Herceptin to the complex, we could actually get a little bit lower resolution reconstruction, but still good enough to be able to model build. And in magenta now you see Herceptin. And what's cool is now if you look at the morphs, the complex basically HER2, HER3 is flexible enough to open up a little bit and accommodate Herceptin binding, as you can very clearly see here. And we're actually quite excited to see we can, now that we have this sort of extra interfaces, basically we have this extra interface with HER3, is there something we can use and utilize for even better binders to HER2, HER3, and better therapeutics. And then what was really interesting, we followed this up with sort of pull down studies. And indeed, we were able to show that, Herceptin binds to HER2, HER3 in a wild type context and pulls down with them. What was actually quite surprising to us was that pertuzumab actually binds right here. So it would be not at the subdomain 4, it would be right at the interface between the heart-shaped dimer. Pertuzumab, we can still get HER2, HER3 heterodimer with pertuzumab bound, as shown here. This is the other antibody. We tried to look at it by negative stain and even cryo, and that never went anywhere. But it surely did not look like a heart-shaped dimer, so we don't quite know what this looks like. But then with the mutant, the S310F mutant, we can see that Herceptin still binds, but pertuzumab doesn't. And we don't quite know if pertuzumab doesn't bind because of the because the S310F is actually right at the interface of the crystal structure with pertuzumab, or is it because S310F stabilizes this heterodimer and pertuzumab does not have a chance to engage? And so, seeing that we can accommodate this Herceptin in the context of HER2, HER3 was really quite interesting to us. So where to next, right? So we got this all of the structures. What's next? Well, obviously, as you've seen, we only really figured out what's going on sort of extracellularly. So we really have no idea what's going on in transmembrane, and we really don't see kinases. And the kinase domains are of very low resolution. We can't really quite place them confidently anywhere. And so if we sort of try to build a model now, this is based on our, based on the other crystal structures and our low resolution fit model based on our Cryo-EM. Here, this is how the heterodimer would look like. But one thing you can immediately see over here is that, and this is actually repeats quite a bit, is that there is definitely, it's not straight down. There's some offset between the extracellular domains and intercellular domains. But another thing you see is that, the amount of membrane you need to have to incorporate sort of all the pieces there is actually quite big. And this is bigger to people who do this. This is bigger than the run-of-the-mill nanodiscs that you utilize. And so I think this really raises the questions of, how far we'll be able to push this in the context of micelles or even the canonical nanodiscs. And I would like to say is that, we definitely spend a lot of time trying to find the kinase domains in our structures. And so we did all kinds of 3D variability analysis. We did all kinds of skip-align classifications, specifically focusing on the kinase domains, because again, realistically, if you have something that's 70 kilodaltons, as stable, we should be able to reconstruct it without having even the extracellular domains. We have in the lab a number of reconstruction of 70 kilodalton things to 3 Armstrong. So it's unclear, something must be going on that we can see it. And we certainly tried adding recentering and subtraction to skip-align classification. So basically anything that we could think of in terms of computationally getting at these in our DDM prepped samples, we could never really get anything. We could sometimes get better blobs, but never really convincing blobs. And so what's going on, we really don't know. I mean, I think it's very interesting because there is, there's still, in the past couple of years, there's been quite a bit of effort on getting this family or other families of RTKs in full-length context. And people sort of gotten to as far as we gotten, where, you often get a very high resolution structure of ECGs, and then everything else is gone. And so is there some, systematic screwing up of redox states of cysteines because extracellular domains need to be oxidized and intercellular domains need to be reduced? Or is it the fact that there's lacking membrane? And so it's unclear at the moment. We've been playing around with both of those things. And so, so one thing we've been really pushing on, and Allison in the lab is really working on this, is going for nanodiscs. And so you can see here that the reconstitutional narrative seems to be pretty efficient in the latest trials by negative stain, as you can see here. One thing she's observed also is, again, going back to this grids where the bare geo grids, when you have graphene oxide from graphene, the way graphene oxide works, you have epoxy groups or just carboxylic acids at the edges, but in the middle, you mostly have epoxy groups. And so it ends up being negatively charged. And so you can use it as a handle, something that Feng developed to attach PEG molecules with a means to make it sort of brush, this PEG brushes with positive charge. And so Allison sees quite a bit different distribution of particles depending on ice thicknesses, if she uses this versus this with the nanodiscs. And, we're starting to get more stuff below, in the micelle region, but, this clearly does not look good. And so we're still not there in terms of getting the reconstructions. And so, but going even further, right? I think, it's one thing to, study these things in isolation, but realistically, the whole point of this thing being phosphorylated is so that it can recruit things downstream of it and build this larger protein complexes and the inner leaflet of the membrane. And so that's sort of where, a lot of other labs efforts has been focused on. And so Komal in the lab has been, as I mentioned, working on this RAF-MEC-ERK pathway, which usually is depicted as a sort of a linear, you can have sort of feedback loops and things like that, but usually that's how it's thought of. But realistically, even if you just look at something like RAF kinase, it has this, RAS binding domain, which binds to RAS when it's bound to GTP. It has a cysteine reach domain, which binds to the membranes loosely. And then you have, of course, the kinase domain. And then you have this 1433 binding sites, which are really cool, where depending, the same 1433, but different sites can either keep the kinase in the off state, as is depicted here. And this was a recent structure solved by EC group, where you can see the N-terminal phosphorylation site and C-terminal phosphorylation site being engaged by the 1433 dimer to keep the kinase in the off state. And what's cool is that also in the off state, you have downstream signaling molecule MEC bound to it. And then upon activation, and this is sort of a imagined state where upon activation, with a say RAS dimer, it's actually being as you'll see, it's conflicting opinions if it's a dimer or an oligomer. But the idea is that this thing should come together. And what's cool is that this 1433 sites, you get a RAP dimer. And now the 1433s are stabilizing the active state by just stabilizing 2 of the C-terminal tails over here. And then the N-terminal sites are being dephosphorylated. And so you have this fundamentally you have this cycle of phosphorylation changes, which follows, which leads to a change in protein-protein interactions and rearrangement of these complexes at the membrane. And trying to understand that, I think, is very exciting. And so Komal has gotten sort of this bit, and we have some classes with 2 MACs, we have some classes with 1 MAC. We see the 1433 engaging this C-terminal sites. And there have been recent structures of this complex published. And what's interesting is even then we see subtle conformational changes because I think we have different mutations in our kinases and different small molecule inhibitors. But what Komal is working quite hard on is trying to push this whole system into being able to fish out using nanodiscs, which are conjugated to RAS, fish out this mammalian prepped RAF activated complexes, and should start getting some headway in that direction. But ultimately, again, these all are large protein complexes which are assembled at the membrane upon receptor tyrosine kinase activation. And so there was this recent paper which was really utilized a lot of MD with some biochemical data, some structural data to build this big Signalzo model, as you can see here. And again, this is just a model, so it's contentious. Some people believe it, some people don't. But the idea is that it's consistent with a lot of data, and they argue that you get this mini filament of RAS on the membrane upon activation, which basically then in its own right recruits downstream RAFs, and then those you can build in based on because this is all based on domain crystal structures. It's still no full length RAFs or BRAF structure, and then 1433s and MAC substrate proteins. And so this is just one vision based on that particular modeling and that biochemistry and the structural biology of what might be happening at the membrane downstream of receptor tyrosine kinases. But realistically, this would be able to incorporate SOS, and SOS is is the exchange factor for RAS, and so it's the downstream of aGFR and GROB2. And so realistically, this most likely happens right in the vicinity of the receptor tyrosine kinases. And so the long-term goal of the lab is to really try to visualize this and capture this in some sort of lipid vesicles, because this is there's some sort of irreducible complexity which we cannot capture by studying individual domains. And with that, I would like to thank everyone. I would like to thank the lab over here. And then again, I would like to underline that all the work which I talked about and all the kinase work in the lab we do together with Natalia. I would like to thank the AIR lab, Dave, for help with all kinds of things, but also Feng specifically for help with all the graphene oxide grids, and our Cryo-EM facilities, David Balti and Glenn Gilbert. And thank you for your attention, and let me know if you have any questions.
Okay, thank you so much, Kliment, for this amazing talk. So I guess now we can open the seminar to questions. So you can, of course, either tap them in the chat box or you can raise your hand. And yeah, so if anyone has a question, raise them now. So Mari, yeah.
Yep, hi. Sorry, I'll just turn my camera on 1 second. Hi,
Hello.
thank you. Thank you so much for the talk. It was really interesting. I'm sorry if this is a bit of a silly question, and also sorry if I misheard. But with your HER2, HER3 dimer, did you say that you truncated a bit at the end? And I just wanted to know, was there a reason why you did that?
Right. So yes, you're absolutely right. So we truncated C-terminal of the kinase domain. I don't remember exactly, but I think we have 20 or 30 amino acid C-terminal of the kinase domains. I think those, so in the field, those are referred to as C-terminal tails. I think they're quite fascinating. A number of them people have looked at by, say, circular dichroism, and they do look unfolded, but they're soluble. So when you prep them, they don't crash out. But some of them are like 250 amino acids long. And so again, as a structural biologist, just, they probably adopt some sort of shapes. And so initially we actually truncated them because we thought that we would take a hit in our, in our yields by having this floppy bit, a bit of a crystallography kind of thinking. For some of these receptors, we actually followed up and done full lengths, and we can get them. Now, do we see the tails? No, we don't see the tails. But in terms of like yields and things like that, we can. And so I think that was sort of just, some of the older school thinking and the construct design. We didn't really have empirical data showing that, having those tails would disrupt them. But, out of abundance of caution, I guess.
Okay, thank you so much.
Sure.
All right, I think Katrina was next.
Hey, Clem, thanks for that lovely talk. I've got a couple of questions. Firstly, I was really interested in the wedging that you talked about with aGFR. Do you see that as there's a sort of continuous distribution of partial sites that can sort of modulate the signaling, or is it that there's full or partial wedging and that's kind of it, it's either one of those two states?
So it's a good question. I would think, so, explicitly with aGFRs, there've been an ERAG structure and AGF structures. There've been sort of the Spitz structures from Drosophila. And so, we only see sort of we've seen so far discrete states. I would much prefer to think of it as a continuum, because again, I think of, a lot of these proteins and this whole pathway, I like to think of it certainly as a computer. And so number of possible accessible states is very important. And so a continuum gives you more states than, two states. But we don't have explicit evidence for that. I would imagine, but we only seen two concrete eGFR states. And again, I want to put it out there that, we're extrapolating from HER2, HER3. So there could be just idiosyncratic things going on with those two proteins, right? And then, it looks similar to the eGFR family, but maybe that is all nonsense, and the eGFR does, the same one thing, and then HER2, HER3. So just as a word of caution. But I would imagine that there would be a continuum. There definitely, ligands usually can be broken up into weak binders and strong binders. So there is that, but they will still have different affinities, right? Yeah. So
my other question is that there's some evidence that they're signaling through higher order oligomers like tetramers for eGFR. Do the structures have any insight into where those additional interfaces might be? And do you think that like the TMs and the kinases having a higher order interface might stabilize them further, and that might be another route to getting that kind of full length structure?
Absolutely, yes. So I feel like fundamentally there's a gap between sort of high resolution structural studies of high resolution understanding of what's going on from say crystallographic and now cryo-EM studies, and mostly been focused on domains, dimers, that sort of units. And then there is a lot of work in cells, often using light microscopy, which just shows that there's all kinds of different oligomers and different receptors within the same family, signals through different oligomers, and they seem to be very important. In our preparations, we see larger things by size exclusion. We tried hard to look at them structurally. We never quite see, anything that is workable. Again, it's size exclusion, so it's not entirely even, convincing if it's larger mass or is it just weird. But I think fundamentally, the micelle is tiny. As you've seen, even a big nanodisk is not enough. And so realistically, that's why I totally think that there is a lot of interesting things which are going on with oligomers. Can we say anything about the interfaces? No. And I think it's just because we're limited to the micelles, because whatever fits in the micelle is kind of what we see.
Thanks, Clem.
Sure.
All right.
Yeah, you'll figure it out.
All right, and I think Isabel had a question too.
Thanks, Clem. That was a great talk, and nice to see you online. I think it was Katrina stole my question, but in terms of, so in the future, are you thinking of moving into Cryo-ET to look at this? There's definitely a lot to do around the oligomer, and this is the same thing with the ephrin receptor. So is it something you're thinking of?
Yes, very much so. Absolutely. So, I think, so Katrina is actually going to be working on it. but yes, I think, so we'll need to figure out exactly how to do it. I think this is a very interesting technical question, because realistically, cryo-ET still, what's the state of the art in cryo-ET? Ribosomes at 3.5 angstroms from the cytosol, right? But these things will be way smaller, way more dynamic, way more heterogeneous, and way lower abundance. And so I think there will be some sort of in vitro vesicle system, which will allow us to bridge sort of cryo-ET world from cells to, in vitro DGM micelle type of things. And, again, things like nanodiscs, maybe large nanodiscs, there are some technologies now which allow you to get bigger nanodiscs, maybe can allow us, at least to see the intracellular domains. That would be already great. But I think just sort of brute force cryo-ET from cells won't quite work yet. Maybe in 15 years it will. But so I think some sort of vesicle type system, liposome type systems is sort of really the way to go. And those will probably be a combo of cryo-ET and single particle, because you sort of can combine, where you collect tomograms and then you do subvolume averaging. And even some of those you can do a single particle on cryo-ET in the same thing.
And in regards to the intracellular domain, so surely there must be some conserved residue that interact, a conservation in residue that will interact somehow with the membrane. Yeah.
Right. So the juxtamembrane domains actually.
Yeah. But even within the kinase domain, I'm sure that there would be a patch.
Yeah, I'm trying to remember if it's, I remember definitely juxtamembrane domains, because juxtamembrane domains wrap around the kinase domains and sort of plop it up there, right? But again, and so, yes, absolutely. Again, I think in DGM, and we know biochemically from DGM extractions is that with DGM, the sample, kinases lose activity and aGFR lose activity over time. So we know it's bad, frankly. It just kind of worked in terms of getting this particular structure. But again, I think it requires a fairly large surface because what we certainly see robustly, as robust as we can see in terms of these blobs, is that, you have the CCGs and you have the ICGs, and they're not on top of each other. So there's always sort of the ICGs are to the side, which I think makes the membrane requirement a little bit larger. So my guess is that we're not seeing it probably because of that. Again, the redox is certainly something I also worry quite a bit about, because it's usually it's, either you have a membrane protein, extracellular protein, or intracellular protein. Here you have both, and trying to figure that out is quite complicated.
Okay, I'll leave a question to someone else. Thanks again, Kliment.
Sure.
All right, so I think Josh is next.
Hi, Kliment. Thanks for the talk. That was great. I was wanting to ask about, you've got a nice structure of 14-3-3 stabilizing proteins, and it's really cool when you have like a natural adapter or a biologically relevant adapter. But has your lab had much success stabilizing things with things like nanobodies or fabs or even Lego bodies? And do you ever do you try chemical cross-linking as well, or what's your feeling about these kind of stabilizers in terms of getting that bigger structures and things like that?
Yeah, no, good question. So cross-linking certainly is run of the mill. So even on these samples, so actually I should have mentioned it, my bad. So these samples, some of the structures were done with some glutaraldehyde cross-linking. We have done them also without cross-linking and seen basically the same thing. So that's why I think it's not super important. But certainly cross-linking is really useful for a number of reasons, right? It's really useful to just stabilize your things, but also I think almost often as a diagnostic too, because often you run the trisacetate gels and you'll see, what do you see? Do you see a smear? Do you see a tight band? Do you see two tight bands and double the size? It can tell you kind of like potentially what the species are. And so cross-linking, I would say, is a huge yes, for sure. Now, for fabs versus nanobodies, not versus, but fabs and nanobodies, so nanobodies for structural work are, so fabs are better, right? Because they're bigger. So nanobodies are tiny. So, and then you're looking at panning campaigns, right? And so if you have a collaborator who's excited and will crank out, that type of reagents for you, absolutely, of course, especially if they're fabs. Now, nanobodies probably will be most useful in, if you can, so nanobodies, again, in terms of just like adding bulk, are useless. But if you can set up a clever screening campaign where you can get allosteric nanobodies to stabilize a state, that's super cool. But those are very difficult campaigns. And so, you need, it's a long-term sort of collaboration with something we talked about with people over here at UCSF. It's just hard. It's a whole own project in its own right. And apparently people in the field, there have been, my understanding, a number of attempts in the field to use sort of a fluorescence-based nanobody screening with some sort of downstream signaling output to try to get biased nanobodies. And that's quite difficult to get robust. And so I think that's sort of the pitfall. On other projects, as I've been involved from this coronavirus, so we definitely got a ton of structures of nanobodies bound to spike CoV-2 protein and as therapeutics. And so, we see those, but not as tools. I think every time as a tool, you need to have a good collaborator who is excited to follow through it, because, it's quite a bit of work.
Thanks, that's really helpful.
Sure.
Right, Naz?
Hi, Klim. Thanks for the great talk. Have you thought of using other systems like saposin lipid nanoparticles for your disk and see whether that would help for getting other?
Yeah, so we talked about doing saposin. Not sure we've actually tried it, to be quite honest, in terms of cutting it out from like a native membrane kind of thing. In the area here, people have had very mixed success, because we sort of surrounded by the Cheng lab on one side and the Stroud lab on the other side. And, there's a number of people who tried it and haven't had, a huge success. It sounded kind of hard. And so that's why we sort of run through the usual, GDN, digitonian, DDM, LMNG kind of thing, and then went to the nanodisk. I also, to be honest, with saposin, I don't quite know how big they can be because, again, I don't remember the sizes. But, looking at sort of initial models, again, whatever our membrane surface will need to be quite large. And so I'm not sure with saposins we can get that large membranes. And that's why I think something like vesicles might be a little bit better.
With saposin, I think maybe like if you want, but the incorporation using nanodisk method might work as well. So you have
not cutting out from
protein.
native, so from purified protein and biobeads, not from, what's the benefit of that over using the usual MSPs in your experience?
Because technically saposin oligomerize around the membrane. So it might, it might give you a bigger disc. Yeah, but I don't know whether it works.
Sounds good. No, we can certainly try because
it's definitely easier than the direct extraction.
I see. Because we definitely talked about going to larger nanodisk, and it's something we're trying to do now. But I think if we can do the same with saposin on an in vitro purified system, it's a good idea. Thank you.
All right, so I guess I have a quick question, if you don't mind, just because you were talking a little bit about the interactions between HER2 and HER3, and you've talked about pi-pi stacking contacts. Have you observed any other stabilizing interactions? I don't know if I've missed it, or also do you think there could be any other studies to actually measure how strongly the complex dimerizes, like any pharmacological?
No, it's a good question. So of course, so the pi stacking was just and that was the interaction specific to that mutant, the cancer mutant. But there's a ton of interactions in between the HER2 and HER3 between the subdomains too all along. They sort of zip up. And going off memory, I mean, I think sort of intermediate buried surface areas are definitely like I think in the aGFR, it's I think more. But there's quite a bit of other interactions between HER2 and HER3 besides that. in terms of measuring them, you know, it's actually interesting. So this is fascinating. So, you know, we know that if you just have the extracellular domains of these two proteins, they don't seem to interact in vitro, at least. And then even purifying totally wild type samples seems to be quite difficult. And so we incorporate these mutations at the intracellular domains to stabilize these dimers and then we get better yields and everything. But we don't see the intracellular domains. And so it's sort of this like puzzle where like we know that we can mutate the intracellular domains to push equilibrium, but we can't quite, you know, we can't quite see those domains themselves. Where I'm going with that is, you know, in terms of measuring the affinities, because this whole thing has been done so much as a sort of a co-purification and very small amounts, it would be quite, I think, difficult. You can probably come up with some sort of sophisticated FRET system, I imagine. But again, even a FRET system would have to probably use, I don't know, fluorescent proteins or something, because there's so many cysteines which you can't mutate them out. You know, things like, yeah, but basically we co-purify them as a complex. And so let's see, you could maybe, what could you do? Maybe you could try to compete it away with something else and then look at the off rate. But again, we haven't done it because it would be hard. A lot of those affinity binding interactions would usually been done, like ERAG versus the GF, have been done on extracellular domains by themselves, which are much easier to work with. Yields are much higher. I think doing this in this context of full-length proteins will be very difficult, and so we haven't actually really done that.
All right, thanks so much, Klim, for your talk today. And on behalf of everyone today, so organizers, audience, and myself, thank you for joining and presenting all the amazing research you've been doing. So thank you everyone as well for participating today. And yeah, see you at the next seminar.
Thanks everyone. Bye. And thanks for hosting.
Thank you. Bye.